All right, today we're going to talk about Chapter 10, Biotechnology and Recombinant DNA. Now, biotechnology, or the use of microorganisms, cells, or cell components to make a product. Foods, antibiotics, vitamins, enzymes, other kinds of medicine. Recombinant DNA, or rDNA technology, is the insertion or modification of genes to produce desired proteins. A vector is another vocab word you'll want to be familiar with. A vector is a self-replicating DNA used to carry the desired gene to a new cell. Plasmids and viruses can be used as vectors. All right, so let's start first with extracting DNA. A source of DNA is needed for manipulation in biotech applications. DNA extraction is the first step in most applications, and it's performed by disrupting the cell wall or membrane by chemical means or mechanical means, and then we collect the cell-free DNA. Next, we use the polymerase chain reaction to make multiple copies of a piece of DNA enzymatically. We call this amplification. Some applications are to amplify DNA to detectable levels. We can sequence DNA or even detect pathogens. So this would be how on a crime scene, if we have a hair with a single hair follicle cell, from a single cell, right, we can use PCR to make millions of copies of that DNA to run identification tests or a paternity analysis, all kinds of different things. PCR is used to target a region that can be used for any gene of interest, usually short portions of the gene, less than 1,000 base pairs. It's remarkably fast. It only takes 2 to 3 hours. Primers are used to selectively replicate only the target region. So when we do PCR, we're not copying the whole genome. We're just copying certain segments of the genome that we're interested in studying. All right, and it provides a 3' end for DNA polymerase to add nucleotides to. So those primers were used to kind of bookend the sections of DNA that we want to replicate. Primers themselves are short sections of DNA that base pair to single-stranded DNA. And then that provides the 3' end for DNA polymerase to add nucleotides onto. PCR is possible due to a very special enzyme called Taq polymerase. It's a heat-stable DNA polymerase from a thermophile bacteria, Thermus aquaticus. And this Thermus aquaticus lives in hot springs in Yellowstone. So it's stable at temperatures up to 95 degrees Celsius. The DNA needs to be heated to about 95 degrees Celsius to break the hydrogen bonds that hold together the rungs of the DNA ladder. That's where the nucleotides bind together. So instead of using DNA helicase like a cell would do, we use heat to denature the double-stranded DNA and break it apart into 2 single strands. DNA extractions and PCR are used in the medical field to confirm diagnoses and identify organisms causing disease. Remember, only a small fraction of bacteria can actually be cultured in the lab. So using these biotech tools to identify things based on their DNA is more precise and accurate, depending on your situation. All right, so Bordetella pertussis is a causative agent for whooping cough. B. pertussis is very difficult to isolate due to its extreme sensitivity to desiccation. It dries out so easily that it's difficult to culture in the lab. Specific primers targeting regions that are only found in Bordetella pertussis are used in PCR, and the presence of these genes that are specific to B. pertussis can confirm that a patient does indeed have pertussis. Some other applications for PCR are the use of restriction enzymes Restriction enzymes allow scientists to cut DNA into fragments, and each enzyme recognizes a very specific 4 to 6 base pair nucleotide sequence. So let's look at some examples. These are selected restriction enzymes used in our DNA technology. So there's enzymes listed here. Their bacterial source, so the different organisms you find them in, and the recognition sequence. And what this is whenever these restriction enzymes like BAM-HI, it always cuts right here whenever it sees this DNA sequence. And for this one down here, ND3, always cuts on this sequence. So what it does is it leaves us sticky ends for recombinant DNA techniques. Or, depending on which one we use, like this one here, Hey3, cuts right across. So instead of leaving sticky ends, it leaves blunt ends. This is a useful tool for DNA fingerprinting. You can uniquely identify individuals on the basis of their fragment lengths. So fragments generated by the restriction enzymes. We mix these enzymes with a sample of DNA, and it's going to cut that individual's DNA up into different sized fragments, and they differ individually. Each individual's DNA is different enough that these enzymes will generate different lengths of fragments between two individuals. So if you had a sample of DNA from a crime scene and you wanted to compare it to a panel of suspects, you could use restriction enzymes to make DNA fingerprints and see if any of your suspects match the DNA you found at the crime scene. All right, so electrophoresis is a way we can separate DNA fragments based on their size. So you would do the restriction enzyme, and then you'd need to be able to find a way to compare the size of those fragments, right? So a DNA sample is loaded into a gel matrix, and an electrical current is applied. Now, smaller fragments travel through the gel faster, and it creates a banding pattern, right? So DNA has an electrical charge. It's attracted to the electrode of the opposite charge, and that electrical charge pulls the DNA through this gel, and it's like Jell-O, right? So if you were going to swim through Jell-O, it would be tough work, right? Because you're pretty big. But you can imagine something much smaller, like, a small fish would have a lot less resistance, right? So smaller fragments travel faster through the gel. And the larger fragments travel more slowly. And what this does is it separates out the DNA fragments so that we end up seeing bands of DNA. All right, so DNA is negatively charged. The negative cathode is black on the electrophoresis rig. The positive anode is red. So we always run from the smoke into the fire. You go from black to red. You want your DNA, which is negative, to run to red. All right, ethidium bromide is used to bind to the DNA fragments so they fluoresce under UV light. So we're able to actually visualize the fragments. So here's a picture of a gel. There are 4 wells, and they're literally like small holes in the gel. So you can load your samples in. So your restriction fragments. And then you would always run a size standard. So a size standard is a product you'd purchase that has DNA fragments of known length so that you can compare and estimate the size of the fragments in your samples. So you load your samples in the well, you turn on your electrophoresis rig, and DNA is going to migrate towards the positive electrode, so to the opposite side of the gel. And you see these bands here closest to the well, these are larger DNA fragments. And the ones that run further to the opposite side, those are the smaller fragments. And then we always run it with a size standard, right? So we know this band right here is 23,000 bases. And this one is 9.4 thousand bases. And we can use this to help estimate the size of the other bands that we see, right? So if we see this band here is just beyond or just smaller than 9.4 thousand bases, you might estimate that it's about 9,000 bases long right here. Here's a better one up close. So here's our ladder, our fragments of known length. And then here's the different samples, right? So these two seem to share an allele here. The brightness is reflective of how much DNA there is at that size. So there's a lot of DNA here at this band. Okay, so let's do an example of a crime case here. So lane 1 is the victim's DNA. Lane 2 is a skin sample from under the victim's fingernails. And then we have 3 possible perpetrators. So lane 3 is one possible perp. Lane 3 is perp 1, lane 4 is perp 2, and lane 5 is perp 3. Do any of these 3 possible perps match the DNA collected from under the victim's fingernails? So that's lane 2. So perp 1 has a similar allele here, a similar fragment, but this one way up here is absent, and this one down here is absent, so it can't be number 3. Suspect number 2, however, shares this band and also this one. So suspect number 2 is a match for the DNA found under the victim's fingernails. And suspect number 5 doesn't match the sample from under the victim's fingernails at all. So 4 looks like a pretty good match for this one. What does the blue box represent? Okay, so this box here, this grayish blue box, represents the electrophoresis gel, right? The black lines are fragments of DNA. The DNA migrated from negative to positive, and the murderer must be suspect number 2 in lane 4. All right, so let's talk about genetic engineering now. Something a little more positive. Genetic engineering in prokaryotes. Look at this. We've isolated the insulin gene, digest a plasmid using a restriction enzyme, and mix with the insulin gene. So we use a restriction enzyme to cut out the insulin gene and leave it with sticky ends. And we're able to mix it with a plasmid and get the plasmid to take it up. So now we've inserted the gene for human insulin into a prokaryotic plasmid. And we can transform this plasmid into a bacterial cell and make a bacteria produce human insulin. The bacterial cells now produce a product, human insulin, that can be harvested and purified. This human insulin, recombinant DNA origin, is structurally identical to the insulin produced by the human pancreas. This human insulin is produced by recombinant DNA technology using E. coli as the production mechanism. All right, probe technology. So DNA probes can be used to locate specific nucleotide sequences of DNA or RNA. They're single-stranded DNA that has a fluorescent dye. So A probe is added to single-stranded DNA that's been affixed to a solid surface. And the probe anneals to the complementary sequence, because if the label it carries, its location can easily be determined. So this is how we can locate where genes are located on a chromosome. FISH, or fluorescence in situ hybridization, is another type of probe technology. It helps us to rapidly identify organisms, bypassing the need to grow them in culture. To identify Mycobacterium tuberculosis in sputum, using specific probes for TB. So we can look at a sample of sputum collected from a patient and use this FISH probe technology to look for Mycobacterium tuberculosis on the microscope. We also have genetically engineered eukaryotes. So transfection is the same thing as transformation, except in eukaryotes, right? So transformation is when DNA is taken in from the environment into bacterial cells. Transfection is when the same thing happens, but in eukaryotic cells. So we have genetically engineered plants, like plants that produce Bt toxin. It's a toxin that's produced by Bacillus thuringiensis. It's toxic to insects, and the gene's been incorporated into corn, cotton, and potatoes. So these crops basically produce their own pesticide. It doesn't have to be sprayed all over the field. The plants themselves produce it. We also have Roundup-ready crops, right? Soybean, corn, cotton, and they resist the effect of Roundup on crops. So if a farmer grows a field of Roundup-ready soybeans, they can spray their field with Roundup. Roundup should kill the weeds, but leave the Roundup-resistant soybeans untouched. Stem cells are another type of biotechnology. Stem cells are special cells that can differentiate into any cell type. They're pluripotent. They have the potential to be a plethora of different things. Stem cells could be used to replace damaged tissues and organs in humans, and they could be used to cure diseases. So it's an active area of research. Embryonic stem cells come from the embryos that haven't completed the process of determination. These cells have great potential. However, embryos have to be destroyed in order to be obtained and worked with them. So there's ethical concerns that go with this as well. Often, embryonic stem cells are stem cells that are donated by parents who've done like IVF, in vitro fertilization. So if a couple goes to a fertility clinic and does IVF treatment, they might end up with 24 fertile embryos, right? And then when that couple decides that they're done having children and they no longer have a use for those embryos, they might choose to donate them to be used for medical research. Okay, adult stem cells and iPS cells. Adult stem cells have partially completed the process of determination. They have less potential, and they're already specific for tissue types like blood or bone, etc. But there's no ethical dilemma in obtaining them because you can get consent from the donors, right? iPS cells are induced pluripotent stem cells. We reprogram mature cells that could be epithelial or fibroblast cells using transfection. And transfected cells contain plasmid genes coding for pluripotency. iPS cells are a newer development and something that looks very promising for stem cell research. All right, so these are some data from a study done by Mr. Pressler at the University of Michigan. So expansion and characterization of human embryonic stem cell derived osteoblast-like cells. So what they have here, these are mouse skulls. the skull D here on the right is a control. So what they did was they cut holes in the skulls of mice and applied their stem cells to the holes to see if they could get new bone growth to fill in the gaps, to repair the damage. And you can see here where they've noted with arrows where there's new cell growth. So here's growth, and then down here, this one did a really great job of filling in the gap with stem cells. All right, so that's it for biotechnology. You should be familiar with the different types of technology, their pros and cons, and how they can be applied.